I'm sure you don't realize it, but your heart beats about 100,000 times a day. The pacemaking system in your chest is what keeps you alive, and quite frankly brought you to life when you were merely 3 weeks old. The circulatory system is the first functional unit in a developing embryo, meaning that the heart is the first functional organ. That being said, I think it is safe to assume that the heart is one of the most important components of human life. But how does it work? Why does it beat? What genes are involved? And where does it start? These are all very important questions that I hope to answer for you in this flipped lecture. After this brief crash course in the development of the heart and heartbeats, I hope you will have a firm understanding about how that red mass in your chest beats and keeps your body going. To begin our journey into the realm of heart development, I think it is important to start with the specification of heart tissue and the heart progenitor cells. A progenitor cell is similar to a stem cell, as it has the potential to differentiate into a specific type of cell, which is known as a target cell. The heart progenitor cells and amniotes gastrula are located on either side of the perimeter streak and the epiblast. The perimeter streak is a structure that establishes bilateral symmetry in the embryos. These group of progenitor cells migrate through the streak and come to form two groups of cells in the lateral plate mesoderm. In the figure, we can see a chick embryo in HH stage 3. Notice how the anterior third of the perimeter streak, excluding Henne's node, is fated to become heart muscle. At AH stage 5, we can see the cardiac progenitor cells have formed in the lateral plate mesoderm on either side of the streak. These two distinct areas are known as the right and left heart field, also known as the cardiogenic mesoderm. The heart itself is made up by a number of different tissues and muscles, but the interesting part is that all these are derived from the same progenitor cell type. Simply put, there seems to be a progenitor cell population that gives rise to the entire circulatory system. These progenitor cells can either give rise to hemangioblasts, which are cells that form blood vessels, or under another set of factors form multipotent cardiac precursor cells. Something that fascinates me about the development of the heart is that the medial-lateral arrangement of these cells will become the anterior-posterior axis of the developing heart tube. The heart cells of the inflow tract, which are fated to develop into left ventricle and atria, structures that receive blood, are located most laterally, while the cells fated to become the base of the aorta, the pulmonary arteries, and the right ventricle are located medially or closest to the perimeter streak. Take a look at the figure showing the cardiac mesoderm and heart tube formation. Like I said before, between stages HH3-7, the cells in the epiblast, which are represented by black dots, ingress through the perimeter streak. During gastrulation, we see a 90-degree shift so that the inflow precursors in blue are located laterally, and the outflow precursors red are located medially. It is important to note that the Purkinje fibers, which coordinate the heartbeat, arise from these two clusters as well. It is at this point in our journey that I meet a few of the main players in heart development. The specification of the cells in the cardiogenic mesoderm is induced by the endoderm adjacent to the heart through two pathways, the BMP and FGF signaling pathways. BMP's role is to promote both heart and blood development, as well as to induce FGF8 synthesis in the endoderm, which appears to be important for the expression of cardiac proteins. To further implicate BMP and FGF8's role in the developing heart, Fujiwara et al. Conducted an experiment that determined that BMP is required independently in two different sites for node morphogenesis and left-right patterning. It was concluded that BMP is necessary for the normal formation of the node and perimeter streak. In order to determine how the lack of BMP affects heart development, Fujiwara and his colleagues generated mouse BMP4 null mutants through intercrossing. Through immunostaining and in situ hybridization, they discovered that FGF8 is expressed in the perimeter streak of wild-type embryos. However, in BMP null mutants, FGF8 expression is strongly reduced. It was also concluded from the study that FGF8 deletion mutants in mouse develop atypical left-right symmetry. This figure shows a stained BMP4 null mutant. We can see from this study alone how important BMP is in the development of the heart, as other genes are highly dependent on it. So how do our heart progenitor cells know where they're supposed to form? We can thank our friend noggin and the WNT proteins. The notochord secretes noggin, which in turn blocks the BMP signaling pathway in the center of the developing embryo, while the WNT proteins that come from the neural tube inhibit heart formation and promote blood formation. The flowchart below should help illustrate the role of BMP, FGF8, noggin, and the WNT proteins in creating so-called boundaries of the cardiogenic mesosome. Once the chick reaches the 18 to 20-hour mark in development, the fated heart cells move from the ectoderm and the endoderm toward the middle of the embryo. The migration of these cells stops once they reach the lateral walls of the anterior gut tube. Specifically in chick embryos, the two cardiac fields that we learned about earlier do not meet until there's a 120-degree rotation of the field with respect to the AP axis. Yeah, that's right. Your heart cells flip and turn until they reach the correct position along this axis. The reason this rotation occurs is so that the cells of the ascending portion of the heart, the outflow tract, are brought rostrally, while the descending portion of the heart, the inflow tract, is brought caudally. This is illustrated in the figure below. The red portion represents the outflow tract, while the blue portion is representative of the inflow tract. The dotted lines in the figure represent the rotation of about 120 degrees. We can also see the development of some mice along the neural tube. The migration of these cells is different in human and mice embryos, as the bilateral fields are brought together to form a horseshoe-shaped field which is known as the cardiac crescent. The formation of the cardiac crescent occurs later in chick embryos at about the 7 semi stage. While the migration of the cardiac precursor cells occur, the posterior region of the developing heart becomes exposed to higher concentrations of retinoic acid. Retinoic acid plays an important role in specifying the cardiac precursor cells become the inflow portion of the heart. The fates of these cells are not determined, but once the precursor comes into contact with retinoic acid, they can start to produce their own retinoic acid, and at this point, their posterior fate is set in stone. We can see in this image that the heart precursor cells in red are exposed to gradually increasing amounts of retinoic acid in orange. We have seen the role retinoic acid plays in birth defects of infants, and this remains true for heart defects. The exposure of embryos to retinoic acid can lead to the expression of atrial tissues at the cost of ventricular tissues. I hope I haven't lost you with all this information. In case you forgot, here's a little checkpoint to refresh your memory on what we have covered thus far on our journey to heartbeats. We have talked about the specification of heart tissue, the migration of precursor cardiac cells, the determination of the AP cardiac domains. We have just begun talking about heart cell differentiation. At this point in our venture, we will further discuss the genes involved in cell differentiation and how this leads to the initial heartbeats. The first transcription factor we will discuss is GATA4. The first emergence of GATA4 is in the precardiac cells of chicks and mice. As these cells arise from the primitive streak, the expression of GATA4 is retained in all these cells that compose the heart fields on either side of the embryo. This transcription factor is important for activating a number of heart-specific genes, and more importantly, it activates the expression of the gene for N-cadherin. N-cadherin is a protein responsible for the fusion of the two heart rudiments into one tube. We will talk about that in greater detail later. Mice that lack the presence of GATA4 have bilateral heart tubes, which is known as cardia bifida pictured on the right. This is where two hearts form, one on each side of the body, and there is a reduced number of cardiac myocytes. The second transcription factor we will discuss is NKX25. NKX25's role is to tell the mesoderm to become heart tissue, as well as activate the synthesis of a number of cardiac transcription factors. Both GATA4 and BMP can induce the synthesis of NKX25, which is expressed in the anterior-most lateral plate mesoderm, seen in the image on the right in the light blue color. As NKX25 and GATA4 work together, they activate the expression of genes encoding for muscle-specific proteins such as the cardiac actin. We will talk more about the role of NKX25 and its relationship to the gene Tin Man at the conclusion of the lecture. The third and final transcription factor I will mention is TBX5. This gene is expressed in both the cardiac fields in chicks and mice embryos, pictured in the dark purple color. An intriguing point about TBX5 is that the ventricular septum is formed at the intersection between cells expressing TBX5 and cells who do not. This transcription factor is integral in the separation of the right and left ventricles. I hope I haven't bored you to death with all these transcription factors and cardiac terminology. Now that we have laid down a solid foundation for some of the genes involved in the role in heart development, we can finally talk about what I know you've all been waiting to hear about, the first heartbeats. Cell differentiation occurs independently in the two heart primordia. As they move towards each other, the ventral splanchnic mesoderm cells of the field begin to express N-cadherin on their apices, distinguishing themselves from the somatic mesodermal cells and fuse together forming an epithelial layer. The fusion of the somatic mesoderm leads to the formation of the precardial cavity, or the sac in which the heart forms. The epithelial layer of the splanchnic mesoderm forms the myocardium, which develops the heart muscles that will pump until the organism dies. The fusion of the two heart primordia occurs at 3 weeks in human gestation and about 29 hours in chick development. At this point, the myocardia unite to form one tube. Pulsations in the heart of the chick begin while the two primordia are still fusing. Heart muscle cells have the amazing ability to develop the innate ability to contract. Scientists have isolated heart cells from a 7-day chick or rat, and these cells will continue to beat while they are in a petri dish. The pulsations in the heart stem from the emergence of the sodium-calcium exchange pump in the muscle cell membrane. According to Wakamoto et al., inhibiting the sodium-calcium exchange pump prevents the heartbeats from starting. Eventually, the rhythm is coordinated by the sinus venosus. Electrical impulses are generated here and then initiate waves of contraction through the tubular heart. This makes it possible for the heart to pump blood even before the valves are complete. So that's it! The heart is beating and beating regularly. As you can see, there are a lot of important steps in the development of the cardiac conduction system, and one misstep along the way can lead to a dysfunctional heart. Now that we know how heartbeats come about, I thought it would be interesting to take a more in-depth look at one of the more crucial genes in heart development in Drosophila, TIMAN. TIMAN is a homeodomain-containing transcription factor, and a gene that we learned about earlier is closely related to TIMAN and serves a similar role in a different species, NKX25. In Drosophila embryos, TIMAN is first expressed in the presumptive mesoderm, pictured in the dark blue in Figure A. After gastrulation, TIMAN expression is restricted to the dorsal portion of the mesoderm, which gives rise to the heart. TIMAN mutants lack a heart and other dorsal mesodermal structures. With that being said, TIMAN is vital for the subdividing mesoderm. Comparatively, in NKX25 knockout mice, the early heart tube forms normally. However, later in development, the heart tube fails to undergo normal looping, and two other transcription factors fail to be expressed in NKX25 mutant hearts. What makes the relationship between NKX25 and TIMAN so interesting to study is that when vertebrate TIMAN-like genes are expressed in transgenic Drosophila, they have the capability of substituting functionally for the Drosophila TIMAN gene, and rescue partially the defects of the TIMAN mutants. How cool is that? A gene in a vertebrate organism can be used to partially restore the functionality of a missing gene in a much less complex insect organism. Phew! Are you tired yet? Me too. I hope today you have all learned a little more than you came here knowing already about embryonic heart development. Heart development is a complex process, and many mechanisms are needed to ensure that the process goes smoothly. Like many of the topics we have learned about this semester, similar genes can serve completely different functions in a different organism. However, in the case of TIMAN and the Drosophila embryo, we see how close related a gene can be to another, despite the vast differences between species. I hope this lecture has made you appreciate the work your heart does, even when you fail to realize it. Thank you, and keep beating on.